Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at monoclonal antibodies. You'll be able to describe how monoclonal antibodies are created, and we'll be able to discuss some of their uses, including their use in pregnancy tests. So have a little think for a moment. Can you list 3 properties about a lymphocyte cell and 3 properties about a tumour cell? 3 things you know about either one of those 2 cells. Well, I hope you said that lymphocytes are part of the immune system. Lymphocytes produce antibodies, and these antibodies will destroy a pathogen by attaching onto the antigens on the pathogen. And for the tumour cells, I hope you notice that tumour cells will divide rapidly by undergoing repeated mitosis, and that tumour cells are cancerous. And that's going to be a big problem in the human body. So what exactly is a monoclonal antibody? Well, a monoclonal antibody is a protein produced by a cloned lymphocyte cell. And these antibodies will bind to a specific antigen, and therefore they're able to target specific chemicals or cells in your body. So how exactly do we make one of these monoclonal antibodies? Well, look carefully at this diagram and see if you can follow the steps and understand exactly what's going on in the production of a monoclonal antibody. Are there any ethical considerations that we should take into account when we are carrying out this procedure? Well, here's that diagram again, only a little bit smaller. So how could we describe what's going on? Well, monoclonal antibodies are produced by using mice. Therefore, we have to follow specific ethical guidelines to minimize any suffering that the mice may feel. So the first thing that we do is we take the mouse and we inject it with a foreign antigen. Now this will be whichever the chemical is that we are interested in, and we want to produce monoclonal antibodies to specifically fit this foreign antigen. So once the mouse has been injected with a foreign antigen, this will stimulate the mouse lymphocyte cells to produce specific antibodies to this foreign antigen. The lymphocyte cells are then collected. You can see that here. The lymphocyte cells are then collected and they're combined with a tumour cell. So here's our tumour cell, and this helps us create this third cell known as a hybridoma cell. The hybridoma cell will have properties of both the lymphocyte cells and the tumour cells. Therefore, the hybridoma cell can divide rapidly, just like the tumour cell, and it can produce antibodies just like the lymphocyte cell. The individual hybridoma cell is then cultured and cloned in the lab to produce many identical cells that can produce a large quantity of the antibody, and these antibodies can then be collected and purified. So we've made our monoclonal antibodies using this process here. So what would we do this for? Why would we do it? What are the uses of these monoclonal antibodies? Well, we can use these monoclonal antibodies to diagnose certain conditions, for example, diagnosing pregnancy on a pregnancy stick. We can use these monoclonal antibodies to detect and identify pathogens in a laboratory test. We can use monoclonal antibodies to measure levels of hormones in a blood in a laboratory test. We can use monoclonal antibodies to research, locate and identify specific molecules in a cell or in a tissue. And this is often carried out by binding a fluorescent molecule to the monoclonal antibody. So the first step would be to bind the fluorescent molecule to the monoclonal antibody. Then secondly, introduce the monoclonal antibody into the tissue. The monoclonal antibody will then bind with its specific antigen. And if you then shine a UV light on, then the molecules will fluoresce to identify where exactly those antigens are in the tissue or in the cell. And this is a similar process that we can use to locate blood clots within the body, and we can also use monoclonal antibodies to treat diseases like cancer. So how exactly would we use a monoclonal antibody to treat cancer? Well, this treatment is known as the magic bullet. You can see here. It's got nothing to do with bullets, but it's known as the magic bullet because we can in effect fire a potent chemical to destroy very specific cancer cells. So monoclonal antibodies can be bound to a range of poisonous substances such as radioactive chemicals, toxic drugs, or chemicals which will stop a cell dividing. So if you bind that specific toxic drug to the monoclonal antibody, then the monoclonal antibody will carry that drug throughout the body, and the monoclonal antibody will only bind with its specific antigen. And in this case, those antigens will be found on the cancer cells. So the monoclonal antibody will deliver its poisonous substance directly to the cancer cells without harming any other cells in the body. And obviously this will destroy the cancer cells and can help us to treat cancers without harming the rest of the tissues in the body. Another use of monoclonal antibodies is in diagnosing pregnancy. And I came across this diagram on an old exam question, and it asks you to explain how monoclonal antibodies can be used in a pregnancy strip. So how would you do that? Well, the answer is below. The monoclonal antibodies are used to identify the pregnancy hormone known as HCG, and that will be found in urine. So when urine is applied to the test strip here at the bottom, point 1, it will be drawn upwards towards the windows at the top of the stick here. In the reaction zone, section 2, the monoclonal antibodies specific to the HCG hormone will bind with the HCG hormone in the urine, and these will form a HCG antibody blue dye complex. When the urine reacts with the results window, up here number 3, this complex will bind with an immobilized HCG antibodies, and it will cause a blue line to appear in the window. When the urine is drawn up and reaches the control window, point 4, any antibodies that did not bind to the HCG will now bind with an immobilized antibodies and cause another blue line to appear. So we will either get a blue line here in the results window indicating pregnancy, or a blue line in the control window indicating no pregnancy, depending on whether the monoclonal antibodies in the pregnancy strip bind with the HCG hormone found in the urine, or they don't bind to this HCG hormone because it's not present in the urine and the person is not pregnant. Well, I hope you've enjoyed this video on monoclonal antibodies. If you have, don't forget to subscribe and comment below to let me know what you have learned. Thank you very much for watching.